Welcome to this video lecture on clinical trials. A clinical trial is a designed experiment for the purpose of comparing an experimental treatment with a control, where the control could be the existing treatment or a placebo. In order for it to be ethical to carry out a clinical trial, the principle of equipoise must apply. That is, it's unknown which treatment will benefit the patient. Because if it is known or suspected which treatment will benefit the patient, then it's not ethical to prescribe the inferior treatment for the patient. Now, a clinical trial needs to be carried out as a designed experiment. It's not enough to do an observational study. It's not enough just to look at the health outcomes of people who do or do not take certain medicines. For example, a certain hormone therapy was believed to be useful against heart disease because the people who took the hormone had better health outcomes than the people who did not take it. However, surprisingly, a clinical trial revealed that the hormone therapy actually increased the risk of heart disease. And the reason why the people who took that hormone had better health outcomes was that they were healthier and more active and less overweight than other people, and they chose to take it, thinking that it would give them yet an extra boost. So we cannot draw conclusions about the efficacy of a drug based on simple observational studies. We need to carry out a designed experiment so that we can clearly identify causes. The first step in carrying out a clinical trial is recruitment. We need to have a sufficient number of participants who will meet our inclusion criteria, so that we can allocate them to taking either the experimental drug or to the control group. Now, this is not as simple as it sounds. Some patients may have a preference for a particular treatment, so they're not willing to be simply assigned to one drug or the other. And participants can also decline to give their consent. They must give informed consent in order to include them in the trial. They may decline to give consent, or they may give their consent and later withdraw their consent. So one way to increase the number of suitable participants is to carry out a multi-centre trial. So subjects or patients from multiple centres are enrolled in the same trial. When we have our list of participants, the next thing we need to do is to assign them to either the control group or the treatment group. And we do this randomly. And this is so that there will be no benefit to either the control group or the treatment group in terms of the type of patients that are allocated to that group. We would use stratified randomization. If there's some inequality in relation to age or gender, we would stratify the population of participants first of all, and then randomly allocate equal numbers from each stratum to the different treatments. We also do this blindly. That is, the participants do not know whether they're on the experimental drug or on the control drug. And this is because a new drug typically generates in people an expectation of better outcomes. And so if some of the participants knew that they were taking the experimental treatment, their expectation of better outcomes could produce better outcomes for them. For the same reason, we typically carry out a trial double-blind so that the experimenters and their physicians who are attending them don't know either unless they need to know. Because they could communicate to the participants an expectation of better outcomes, and this behaviour on the part of the experimenters could affect the participants and cause them to produce better outcomes. When the participants have taken the drug, then of course afterwards, towards the end point, they need to be followed up. And that's not as simple as it sounds, because time passes on a clinical trial. And some people can drop out, choose to drop out, or they can simply be lost to follow up. They might move address or move country. Now, if they drop out at random, that would be fine, because then we could just ignore the missing data. But we cannot assume that participants drop out at random. They could drop out because the treatment they took had some negative side effect. And so where someone drops out, we need to perhaps use an imputation strategy to impute the missing data by replacing the missing value with a value taken from another participant who had similar characteristics or in some other way. The next step in a clinical trial is to decide on the data set for analysis. Now, the data set we typically use is the intention to treat data set. That is, when we analyse the data at the end of a clinical trial, strange though it may seem, the complete data set that arises for all of the original participants should be used in the analysis, even if some of these participants did not comply with the protocol that was prescribed, and even if some of them never took the drug they were allocated. And there are a couple of reasons for taking this approach. One is that non-compliance can be related to side effects. And the second reason is that compliant participants, regardless of whether they're in the control group or the treatment group, compliant participants tend to have better outcomes than non-compliant participants. So if we simply remove the non-compliant participants from one group, we will unbalance the design. So the most reliable way to analyse the data is by using the intention to treat dataset. We can follow that up with the as treated dataset, taking account of the drug which the participants actually took, or the per protocol dataset, which pays attention to the patients who took the drug exactly as prescribed. Now, the purpose of a clinical trial is typically to establish which treatment is superior. And for that reason, we actually carry out a two-tailed rather than a one-tailed test, because we don't know in advance which treatment will be superior, even if we think we know. So it could be the experimental drug or it could be the control drug. Sometimes a clinical trial has the purpose to demonstrate non-inferiority. Let's say we wish to replace an invasive treatment with a treatment which is less invasive or less costly, but which is believed to be as good as the more invasive treatment. Well, then we simply need to establish non-inferiority. And so this could give rise to a one-tailed test. We just want to show that the experimental drug is no worse than the existing treatment. In order to determine the sample size required for the clinical trial, we first of all need to get a clinician to identify what is the magnitude of the therapeutic effect that is required. And that's a clinical judgment. That's not a statistical task. That's a clinical judgment. So how big of a difference do we require in blood pressure reduction or whatever other measurement we may be making on the participants? And then when we know the magnitude of the effect that's required, the difference, we carry out the power and sample size calculations in the usual way, as previously described in Chapter 5. Now, the endpoint from a clinical trial could give rise to different types of outcomes. If it's a discrete response variable, if it's a categorical outcome, a yes-no type outcome, or another categorical outcome, we can use contingency tables, and these were also presented previously in Chapter 5. So, for example, let's say we have an intervention for the remission of psoriasis. And so some patients are given this intervention and some are not. And so we have this randomized trial that gives these outcomes. We construct a contingency table and we carry out the contingency table calculations with the observed frequencies, the expected frequencies, the chi-square contributions in the usual way. On other occasions, the endpoint may give rise to a measured variable, such as reduction in blood pressure. And so here we have two alternative drugs being considered, and the reduction in blood pressure for the patients on each drug are recorded. This could be analysed using a two-sample t-test, which was also described in Chapter 5. The t-statistic, the degrees of freedom, we test on a hypothesis, and in this case we see that drug B provided a greater mean reduction. Sometimes the endpoint consists of survival data, the time to an event, such as a heart attack or death or an onset of some condition. Now, it could be that at the end of the follow-up period, some participants have not experienced this event, and so their survival times are right-censored. We can analyse survival data using the Kaplan-Meier method, and it's illustrated here on a survival plot. And we can use a log-rank test to compare the two groups. So the null hypothesis says that the two groups have identical survival distributions. In this example, the p-value is less than 5%. So we say that the survival distributions are different. We notice here that the treatment is associated with longer survival times than the placebo. You can read more about this in the textbook in section 5G.